Okay, we are still in chapter 6, the next chapter or the next topic, meiosis. Okay, so meiosis is the process of cell division that occur is not everywhere, it's just in the reproductive organ only. So where is the reproductive organs in human? Okay, yes, okay, the ovary and the testes. Now these two organs are so special because their function is to produce gametes. And then the gametes, as you know, in sexual intercourse, okay, or during sexual reproduction, the gamete have to fuse with another gamete. That means the male and the female gamete have to fuse together, meaning they plus up together. So that's why meiosis here is quite important, okay, because they have to undergo, the cell or the nucleus have to undergo meiosis so that the nucleus will contain only half the number of chromosomes. If we use human as example, so human have 46. So in order to have this sexual reproduction, okay, the gamete that produced must have only 23. So how to cut off, okay, or make half the chromosome? Okay, this is the process. Okay, the meiosis is the one. Okay, is the cell division. Okay, so during meiosis or during the process of gametogenesis, okay, meiosis happen lah. Okay, so meiosis is incorporate in gametogenesis. Okay, this one you will see again, okay, or we will understand in detail, okay, in the last chapter, okay, chapter 15. So for now, okay, just know the name gametogenesis. So genesis means build or make up, okay. Gameto means gamete, so the process of making the gamete, okay. And then in the process meiosis happens, okay. Now let's see. So meiosis will occur in the testes, of course, this is the organ, okay. Ovary is a female one, okay, in animals and also human. So we focus on humans, okay, but most of the animals also undergo the same thing, okay. So meiosis is divided into two stages, okay, meiosis 1 and meiosis 2. Okay, before today, okay, you have learned mitosis. Okay, mitosis also have stages, okay, but it doesn't divide into mitosis 1 and 2. They straight away into 4 stages. Okay, so for meiosis, they divide into 2 stages first, then every stages got 4 more stage. Okay, so 4 more stages. Okay, for both process, the chromosome replicate only once during interphase. So what is this meant? Okay, so meaning, okay, you still remember the cycle, the cell cycle. Okay, so cell cycle start with interphase, then in mitosis you have mitotic phase, and then cytokinesis, then you get your 2 daughter cells, which is identical. But for meiosis, okay, the process, it will be interphase, okay, meiosis 1, meiosis 2, cytokinesis. Okay, so there is no more interphase between meiosis 1 and meiosis 2. Understand? Later we also will come again with the cell cycle. Now, why we need meiosis? Now, remember the pen here, so means you have to write inside your notes. So if you see just now previously, okay, also with the pen one, you have to write, okay. So the need of meiosis, first thing, to ensure the diploid chromosome number is always maintained, okay. So the word is maintain, okay. Maintain means not change from one generation, from, I mean, if we see in human, yeah, from your great parents until now, us, we still remain the 46 chromosome, okay. Our number of chromosome never add on or disappeared, okay. So how to maintain that? Okay, that is why we need meiosis, okay. So along the discussion here, then you will see lah, okay, how the number is maintained. Actually, if you think a bit, it's logic lah, okay. I mean, you do quite, I mean, you have, if you think a little bit, you will have the idea on how it's being maintained, okay. Now, meiosis also produce genetic variation, okay, among the same species. So this is also happen during the process, okay, during the process of meiosis, okay. We can see there is a genetic variation. We will mention this again, okay, later on when we explain about the stages, okay. Now, this is how we explain how it's being maintained, okay, or why meiosis is so important for the animals or the humans, okay, who have this sexual reproduction. So every time or any animals or any organism, in order to have sexual reproduction, meiosis must occur first, okay, meaning to say before, if the animal or the plant, okay, they undergo sexual reproduction, okay, they must produce gamete through meiosis, okay. So you can see here we have haploid egg, which is the ovum, okay, and then haploid sperm, okay. The word haploid you have learned, before today. Mitosis you see diploid, okay. Haploid means half, okay, so half of the chromosome, half plus half always equals 1. So after they fertilize, they become 1 again, okay. So every organism, if they undergo sexual, you will see if this is the mother represent by blue, half from the mother is blue, another half is red, okay, from the father. So when they combine and become the offspring, okay, or the anak lah, okay, the son or the daughter, whatever, they will have their parents what we call chromosome. That means half of the chromosome, in order to become 46 again, okay, half from the father, okay, the other half from the mother. So in biology, we will use the term paternal chromosome and maternal maternal chromosome, okay. So and then the word homolog here, later you will see homologous chromosome. So even though they get half from father, half from mother, okay, the 23 and the other 23 from the father will be exact, not exact, is quite similar lah, okay. That's why we use the word homolog, okay, similar in a terms, biological terms, okay. This one also we will see again, okay, what is meant by homologous. So for now, for this slide, you just know that, okay, haploid from the ovum, this one shows the ovum, and then haploid from the sperm. When they combine, half plus half equals to 1. So during fertilization, the formation of the offspring, okay, will carry both chromosome, half from the mother, half from the father, okay. The father one is called, okay, we use the term paternal, the mother is using the term maternal, okay. So then, okay, this one is just another explanation. So because meiosis is very important, it is a key factor that generate genetic diversity, okay, and then also they reduce the number of chromosomes, so that the number of chromosomes will not double, okay, based on the previous slide, okay. If I do not, or if the organism do not make it half, every generation the chromosome number is being double, okay. So this is higher, the life cycle of the higher life form. So higher life form mean this is more to extra knowledge lah, okay. Different animal, different type of organism, okay, they have their different life cycle, okay. So this one is just to read out, or you can just to know only, okay. So what we can see here, yeah, the human, okay, I need you to see here again, okay. Human, you have ovum, testis, they produce the gamete through meiosis, can see. So the meiosis is here, okay. So meiosis, so female or male both will have meiosis to produce their gamete. So one sperm here, another one will be the ovum, it become haploid. So half plus half, fertilization means it join up and then it becomes diploid again, see. So after it becomes diploid, every process or every cell division happen after the fertilization, it will be mitosis until the boy or the baby get puberty, so become, okay, can produce gamete. Then again the cycle goes on, okay. So yeah, I need to explain on the homologous, right? So this is what is meant by homologous. So chromosome are match, okay, match in a homologous pair, okay. What is meant by pair? Okay, this is one chromosome, this is another one, okay. If we say just now paternal maternal, okay, this one from father, maybe mother, okay, another one will be from the father, okay, one from mother, one from father. And then even though they come from the sperm and the ovum, okay, they are always similar. So when they are similar, okay, we call them homologous. Okay, homologous. Okay, you see here, if I zoom in, or we zoom in, okay, every parts of the chromosome there will be a location, okay, or we call it locus. So all the locus here, what we call the address, okay, is on top of this chromosome. They have different location, so this location will bring different type of gene. Okay, so every location in biology we use locus, okay, even if every location here will carry different gene. But when we talk about homologous pair, the gene that is being carried over here, the green one, you see, and the one that is homologous to this one, okay, the one number two lah, I put like this. Okay, they are actually same, meaning if this is the color of the eyes, okay, if this one they bring the genes for the color of the eyes, this is also the color of the eyes. Okay, so maybe this is brown color eyes, and then this one is black color. Black, I put a small b lah. Okay, represent black. Okay, so big blue color, small black color. Okay, so if the human or the baby or the offspring in their body, they receive the inheritance, okay, blue color from the mother, they receive black color from the father. Now what is the color of the eyes? Okay, that one we do not know. Okay, that one we do not know until you learn genetic in Form 5. Okay, so but now you have to understand. Okay, homologous means every time when you say homologous pair, they have the same location, their length is the same, and then their location or the locus will carry the same gene, okay, the same characteristic genes. Okay, so but just different version means like this lah, one blue, one black. Okay, that is meant by different version, but they both of them must be the eye color lah. And then go down here, this line maybe the skin color, and then down somewhere maybe the height, and so on. So we have so many characteristics in the body. Some they are obvious for you. Okay, we can see. Okay, we can see. Some not obvious. Okay, even the amount of enzyme, hormones, all are kept inside the chromosome. Okay, I use the eye color because it's quite easy to understand. Okay, now this is another way of they draw out. Okay, oh, just now my drawing no good, right? So this one I got it from internet. I got it so that is more clear here. Okay, so if this is the red one from the father and mother, then the blue one from the father. Okay, so the C, you see here, they will carry the same code color gene. You see, this is color gene maybe. Okay, the color for the fur. Okay, the fur of the rats. Okay, this one they use the rats as a sample. Then at this part, okay, this is the gene for the eye color. Okay, so when they undergo meiosis, okay, they being separated. Okay, so when they being separated, so maybe this part or the part here will be transferred. So what I want you to see is just homologous chromosome. Okay, homologous chromosome means two chromosomes which are similar in the size and the shape. Okay, this is the one that we show in meiosis. Remember, this one we learn, or I show you during meiosis. Okay, I say this is one chromosome, two chromosome. They are together, first pair. Okay, so after meiosis, when they want to divide, okay, when the chromosome during interphase, they will duplicate themselves. Remember, duplicate. So when they duplicate, they become X like this. Okay, so the X left and right we call sister chromatic. They are actually sister, means they are photocopy of each other. Understand? It's not become 1, 2, 3, 4 chromosome. It's just 2, but each of them have photocopy of themselves. Okay, so still 1, 2. Okay, 1, 2 chromosome. Here also 1, 2. So 1 pair, 2 pair, second until the last 22, and we mentioned the sex chromosome, the last pair. Okay, similarly, after an interphase, they will become like this lah. Okay, so that is before we can explain meiosis, you have to understand that. Okay, so now we see meiosis. Okay, so as I said, okay, meiosis we have 4 stages. Mitosis we have 4 stages. Okay, meiosis also 4, but just that this 4 is repeated. Okay, that means in 2 rounds. Okay, 2 rounds. So after it repeat in 2 rounds, it become 8 stages lah. Okay, so 8 stages. So if you see meiosis, then become 2, and then some more meiosis. Okay, meiosis 1, meiosis 2. Okay, so the name also the same: prophase, metaphase, anaphase, telophase. How to differentiate them? We put the Roman letter or Roman number there. Okay, prophase 1 means it's for meiosis 1, metaphase 1 for meiosis 1, anaphase 1. So when you see 2 means it's meiosis 2. If you see 1 means it's meiosis 1. So when you explain about meiosis, this include this Roman number, yeah. Okay, so that one you don't need to write. This one you have to write inside your book lah. Okay, so meiosis got 2. Okay, meiosis wise, 1 comprise of prophase, metaphase, anaphase 1 and telophase, and then you have meiosis 2 comprise of prophase and so on. So this one you write in your notes lah, shorter. Okay, now we have this short video, so we can watch a while. Okay, let's see. Why never start?
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell.
Okay, so you just listen first. This beginning is what I have explained. At the back, okay, is all the meiosis process that you're going to learn. So you just have a rough idea, okay, and then when we go on, we can go a little bit faster, and then you have the pictures lah, okay, we can visualize. Okay, so let's see.
For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis 1 and meiosis 2. Although the processes of mitosis and meiosis look similar, there are a few major differences.
Okay, so there are a few major differences. Okay, the subtitle is down there. So one obvious, okay, later lah. Okay, so you can see, so meiosis, so here same, okay, almost same, okay, but the way of the chromosome is being separated is different. Okay, later after you understand, this mitosis actually happened at meiosis 2. Meiosis 2 is exactly the same like mitosis. Okay, meiosis 1 is the stage that breaks the chromosome into 2. I mean, reduce the number of chromosome into half. Meiosis 1 reduce into half. So meiosis 2 just to duplicate them only. Okay, to make them into 4 lah. Okay, so that's why in meiosis 1, 2n become n. Meiosis 2 start with n, still remain n. So when we say that, okay, mitosis and meiosis same lah, because mitosis you start with 2n, end of the day also 2n. Okay, so that means we look at the number of chromosome. It's not 2n or the n. Okay, this one starting got 46. End of the day after mitosis also 46. Okay, meiosis 2 starting 23, and also 23, never change. Okay, but meiosis 1 starting with 46. When the end of meiosis 1, you have 23. So meiosis 1 is not the same as mitosis, but meiosis 2 is exactly same as mitosis. Okay, so we just proceed lah.
One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, Meiosis begins after a cell has successfully completed the G1.
See, cell cycle, huh? As I said, okay? Interphase, G1S, G2. After that, meiosis 1, cytokinesis, meiosis 2. So there is no more interphase in between, okay? Straight away, meiosis 2, then cytokinesis, okay?
S and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis. Up to this point, the cell looks similar to mitosis. But 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic.
Can you see here? Just now, here is purple, down is blue, okay? So after that, okay, after crossing over, okay, just now they come close, they call synapsis, huh? The video a little bit of lag, okay? Then when they exchange material, so this one they change material, right? Okay, they can change many places, so just that we give one example, they only exchange here lah, okay? This side, okay, this locus. They can actually change any locus along here at the same time, not only one, okay? It can be anywhere, okay? So when they come close together, they exchange genetic materials. So that is that create genetic variation.
Diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fiber.
Okay, we don't use pro-metaphase. We always combine them. Okay, metaphase and pro-metaphase combine lah. Meaning, if you see your book, okay, your textbook, okay, we combine, make it easier. Okay, the spindle fiber attached to the centromere, the spindle fiber form, nuclear membrane disappeared or disintegrate. Okay, that is for prophase or prophase. Okay, prophase.
By their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the
Okay, you can see the difference here. In mitosis, they don't have homologous. So this yellow one, the spindle fiber, okay, in mitosis, they attach to one sister chromatic, one like that. But in meiosis, okay, they attach to one homologous down there. This another chromosome. That means one pair, right? So every chromosome will have one spindle fiber. That means they one spindle fiber attached to one chromosome. Mitosis, one spindle fiber attached to one sister chromatic. Got it? Okay, it's different. Meiosis, one every spindle fiber attached to one chromosome. Okay, there are 2 chromosomes here. This one only 1 chromosome. Mitosis only have 1 chromosome. Meiosis have 2 chromosomes. So meiosis, every chromosome will attached by one spindle fiber. For mitosis, just 1 chromosome, so they attach to sister chromatic. Okay, this is the key difference here lah. Okay, how it become half. Okay, the number of chromosomes become half.
kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to
Okay, here example, how many chromosomes show? 6, nah? There are 6 chromosomes here. Okay, 3 pair, 6 chromosomes. Okay, if meiosis, nah, there's only 1, 2, 3 lah. Okay, they don't have blue and purple different. Okay, they are just a line, one line. Okay, you can refer back to the meiosis videos. Okay.
Independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is 2 haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity.
Okay, we will see this on creating genetic diversity. Yeah, creating genetic diversity. You can see here. Okay, how the human or animals, okay, that undergo sexual reproduction, they create genetic diversity. There are 3 major events that happen. Okay, so one of them is the synapsis or during crossing over. Okay, you just recall this. Okay, or you just you can repeat the videos. Okay.
This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process...
Okay, you can see after they sort, it's different. This one is purple, purple, blue. Okay, this one is purple, purple, blue, but it's the shorter one. Okay, okay, so this one is blue, blue, then a little bit of purple. So they're actually different. Okay, you have to slowly think about it. If you cannot, you ask. Okay, you can private message. Okay, so this is another way because of the random assortment. Okay, independent assortment. So this is Mendel laws. Okay, in chapter, you will learn this again in genetic. But now, if you can understand, it's good. Okay, so how is create diversity? Okay, genetic diversity. So we proceed. There's a bit only.
Process called independent assortment produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
Okay, so it's quite clear lah. So I like this video because it's very clear. Okay, you can replay again and again. Okay, after you read through anything that you're not sure, you don't want to see again, you can just repeat. Okay, so it's good lah. I mean, I like the videos very much. Okay, so
Have
it
you ever wondered
helps.
why children
Okay, now next continue. Okay, you see the pen, so this is you have to write lah. Okay, you have to write in your notes. So meiosis 1, 4 stages. Okay, prophase 1, metaphase 1, where is the 1 okay, and telophase 1, please add on all got 1. Okay, 1, 1, 1, then 1. Okay, during preceding interphase, that means before, okay, it will replicate to form sister chromatids. So this one is almost the same like mitosis. Okay, the identical and join in the centromere, everything is the same. Okay, now this is extra knowledge. Okay, yeah, so we need to know what is hemnasis and crossing over. I think the book also have, yeah, this one. Okay, prophase 1. So after meiosis, so the first stage in meiosis 1 will be prophase 1. So chromatin shorten, okay, thickens. You can compare with your mitosis, prophase in mitosis. Okay, visible. Now this is different. So the pairing of the homologous chromosome, the process is known as synapsis to form bivalent or tetrad. This one, if you can understand, you just write. If you cannot remember, just the pairing of the homologous chromosome in the process of synapsis is very good enough already. Okay, so then the during the synapsis, yeah, crossing over happen. Okay, crossing over means the exchange of genetic materials, okay, between the non-identical chromatic. Non-identical chromatic means the homologous pair. Crossing over produce combination of gene. The point where they cross over is known as chiasma. Okay, chiasma at the end of prophase, and then disappear. This one same as mitosis lah. Okay, and then it move to opposite side. Okay, this one you have to do reading and then memorizing. Okay, no other way. Okay, after you understand, you start to memorize the keywords and so on. Okay, so this one just a little bit on crossing over increase. I mean how the crossing over makes the genetic variation. Okay, now here is what you can say. Okay, this is centromere, right? Centromere. Okay, so these are the chromosome. This is called non-identical. So one is red, one is blue. So when they cross over, they can cross over here, cross over here. See, many point of crossing. Every point is called the chiasma. So here we have chiasma. Here also the chiasma. In between the blue and the red. Okay. This one is between blue. So we don't call them chiasma. It's centromere. Okay. Between blue and the red. So every time when they come over, I mean during synapsis, they have many point of chiasma, and then they can have many point of crossing over. Okay. So then metaphase 1, of course, equatorial plane or the metaphase plate. Okay. Same thing. All right. Like mitosis. Okay. So you can see here. Okay. Yeah. Move. You see. After the synapsis, they move to the metaphase plate. Okay, metaphase plate. So spindle fiber attached to the centromere of the chromosome. Okay, they are one pair. So every one of the chromosome, each one, okay, will get the will attach to the spindle fiber. So when they pull, yeah, you see the whole chromosome move. Okay, this one, two chromosome. Okay, you can go and compare with mitosis. Okay, so if you see here, there is some blue among the red. Okay, that shows that crossing over happened. So whenever you see something like this in exam or in the question, there is crossing over, is meiosis already lah. Just whether it's meiosis 1 or meiosis 2. Okay, now then you have telophase. Okay, plasma membrane constricted. Okay, and then so on. You just read up. Okay, this one you have to write down. You can do short form or short summarize it. Okay, based on your understanding. Okay, but the key point must be there lah. Okay, then at the end the slide there will be a key point. Okay, metaphase 1, this one extra. Okay, so this one meiosis 2 lah. So just now meiosis 1 already you have 2 haploid daughter cells. So meiosis 2 will start with 2 haploid daughter cells. So you see there's the chromosome. It's no more homologous. It's all different length. Okay. so very similar to mitosis. So again, the nuclear membrane disappear, the spindle fiber form. Okay, then here you see it attached to the sister chromatid. Each sister chromatid will be separated here. Okay, the sister chromatid. See the word here? Sister chromatid is being separated. Just now is homologous chromosome is being separated. Okay, so if you want to draw better, okay, if you draw you can know better. Okay, then end of the day we have 4 haploid. Okay, 4 haploid daughter cells. Okay, so that basically on meiosis. Okay, let me see. All those notes that without the pen is just extra. Okay, so this is a slide on genetic variability. So in the video just now also, this one is just in word lah. Okay, in word how meiosis create genetic variable. Okay, so first thing is the genetic combination. So during crossing over, okay, this is what is meant. Okay, so crossover, blue, a little bit of yellow, so you have genetic combine. Okay, crossing over, see, it can be like this, up and down, but it must be between the homologous chromosome, the non-identical sister chromatid. Okay, it cannot be between the blue and the blue. If the blue and blue change, they are actually photocopy of each other only. Okay, no matter how they change, also same. Okay, remember the blue is the photocopy. Photocopy means the duplicate of the chromosome. Okay, so it's during interphase, one become two. Okay, so that's why they are same color blue. Okay, and same length. Okay, so if they change among here, okay, crossing over among the blue, nothing. Okay, it's still the same thing. Okay, I mean from here, from here change to here, same. No worry, it happened, but it's still same. So we wanna create any difference, so we ignore it lah. But when they cross over between the red and the blue, that is for genetic variation. Now, secondly, you have independent assortment. Okay, so what is meant by independent? Maybe just now you're not sure, right? So we see there are four combination here. 1, 2, 3, 4. Okay, so let's say just now all the blue one side, all the yellow one side. Okay, it can be these are all together 6 chromosome. 3 pair 6. Okay, so it can be 1, 2, 3. The third one is the yellow one arranged because they are arranged according to their length. It's not according to their colors. Understand? So colors, it shows that from father and the mother. Remember? Okay. So it can be one side all like this or one side like. Remember the 2 power of this one got 6, right? So it will have 2 power of 6 combination. Okay. 2 power of 6. You can use calculator lah. I think 1 to 8 is it? Okay. So 8, 60, 16, 64, 1 to 8. Okay, so you just do your calculation. Okay, so that is meant by random independent assortment. So it can arrange themselves, and then you see when they divide, you see the dotted line means after metaphase into anaphase, right? So anaphase means this haploid will become all blue. This one become more all yellow. That's why sometimes your sibling are more to your like your grandfather, or sometimes your sibling are more to the look like father or look like mother, or doesn't look like anyone. Okay, because it's like mixed like this, random. Okay, it become like the don't know who. Okay, I mean own self look lah. Okay, so that is all because of independent assortment. So this is meant by independent. How the homologous can be arranged, the probability of being arranged. Okay, now this is another picture if you cannot understand that. Okay, so this is 1, 2, 3, 4 chromosomes. Sometimes they arrange like red, blue, blue, but sometimes it's red, blue. Okay, so when they divide, they become like, oh, this one of blue or red, but this one got combination, you see. Okay, so that is what we call assortment. Okay, so after this, okay, you have to do your comparison, and then you contrast between your meiosis and mitosis. Okay, so compare. After that, you have to do comparison. Is your way of thinking. Okay, anything when you meet something that you don't understand, you ask on the spot. Okay, I cannot force you to understand. You have to sit down and understand it yourself. Okay, so after that, you do 6.3 formative exercise. Okay, so I think the rest will be on the issue. Okay, so far any problem? Okay, mitosis, meiosis, okay, no problem. Okay, So we will see 6.4. Continue just straight to the point. Okay, so the issues. Okay, the issues. Issues in cell division means what happens if the mitosis is not controlled. Okay, what happens when the meiosis is not controlled? So for mitosis, it will form tumor. You can actually read from your textbook lah. Okay, I got it from the textbook. So tumor is actually the flesh. Okay, I mean when the mitosis happen to is not under control. Yeah. Okay, we will have too much of cells. Okay, the extra cells they clump together and we call them a tumor. So tumor can be divided into two lah. One is benign, one is malignant. Okay, malignant. Malignant means it's not good. Okay, it's a cancer cells. Okay, but if benign, okay, then it's okay. It can be removed through surgery. So you can just read out all the causes and so on. So what I need you to do, okay, this is breast cancer, right? So that means if the tumor start to grow and then it can spread into the blood vessel and then it's sent to others, then this is called the malignant tumor, okay, when it can spread. But if it's just a tumor, it doesn't spread, then one is benign, okay? So you have to read, okay? You have to read. After that, you have assignment, okay? So for meiosis, in your textbook, only one example given lah for now, okay? In Form 5, you will see more, okay? So uncontrolled meiosis, you have Down syndrome. So what is Down syndrome? The spindle fiber fail to function or fail to separate, okay? Separate the chromosome, okay? So as a result, the chromosome doesn't separate. So you will see that some of the gamete will have 22, some of the gamete will get 24. So if the 22, with the 24 one, okay, fertilized with another 23, you will have 47 chromosomes. So it will cause the trisomy 21, okay, trisomy or the Down syndrome. So this one, trisomy 21, let me see the picture here, okay. So let's say this is a normal meiosis, okay, normal human, okay, normal, I mean we will call it meiosis, okay. This one is okay, I mean good meiosis. It produced 223 haploid, okay, but there is another part on the partner, okay. If this is a male, this is a female lah, okay. So the partner, something happened during meiosis, okay, one of the chromosome doesn't separate. So if the chromosome happen to be the 21 or the pair 21, remember we have 22 pair plus the sex chromosome. So if you see here, first pair, second pair, and so on, and then 21. So the 21 pair, one never separate. So and then in so coincident also that ovum or that sperm is being fertilized, then the children, the offspring that's being produced, the child that's being produced will carry 47. So we call this condition, okay, Down syndrome. Okay, there are many types of uncontrolled meiosis disease. Okay, this is one of the examples first. Okay, then yeah, your assignment on the band 6, for band 6 now, you have to do a short videos, okay, on the uncontrolled mitosis and meiosis. Okay, you don't need to explain what is mitosis and meiosis, but I need you to put in what is meant by uncontrolled mitosis, uncontrolled meiosis. If you can put in extra information, that's better, that's good, okay. You've got more information, good, then you just add in, explain. I want your narration inside, I mean I want your voice inside the videos, okay, not showing only, okay. Put some music, put some your sound, your voice. You can record your voice and put inside, explain a bit, okay. It will be your assignment for your band 6, okay. So then end of the day, you will do your formative practice as usual, okay. So I think also you do summative practice, okay. I think that's it, okay. The rest will be extra notes, okay. So any question you ask, okay.